Hello, I'm Frank Helle, and I am a chemistry and biology teacher at Bergen Katedralskole in Bergen, Norway. And now I'm going through May 2016 biology exam standard level, paper 1. And the first question is, which molecule is a sugar? And the easiest way to recognize a sugar is looking for the hydroxyl or OH groups. So we see this is a pentose, 1, 2, 3, 4, 5 carbons, and it would typically appear in RNA, in the backbone of RNA, since it has an OH group in the deoxy position. DNA would not have this. B is a lipid, maybe a steroid hormone, maybe a cholesterol. I don't recognize specifically what this one is, but it is a lipid because it has only CH3. So this edge here, for example, is CH2 with a bond to other carbons here. So we see that it's mostly or only carbon. C is an amino acid with the central carbon and interchangeable R group. And D is a fatty acid. Here again, the acid group. But A was the correct answer. 2. Why is sweat a good coolant for the body? And sweat contains water. And water contains hydrogen bonds. So for example, here we have a water molecule, and this hydrogen is a little bit positive. You show that with this strange sign with a plus, and it makes kind of this bond called a hydrogen bond, H bond, that's short for hydrogen bond, to this oxygen in another water molecule. So the hydrogen bond is between water molecules. And what happens with evaporation is that the oxygen or the water molecules, they separate. So here this water molecule is floating around here, totally independent from this water molecule. There is no bond between them. And to break bonds in chemistry, you need heat. And this heat has to be supplied in order to break this bond. And so when this heat leaves your body, it helps cool. It helps you cool down, so it's a coolant. Number 3, which is an effect of protein denaturation? So A is the order of amino acids is changed. No, like amino acids are bound by peptide bonds and they don't easily change position or order. B, the bonds between amino acids are broken by condensation. So this alternative starts promising because there are indeed bonds in proteins between different parts of the chain. For example, a sulfide bridge, but they are not broken by condensation. Condensation is the process where you form bonds. Parts of the protein become linked together by hydrolysis. So denaturation would be the opposite of what C starts describing. But linking proteins together would not happen by hydrolysis because hydrolysis in breaking stuff up. So the correct alternative here is D. The three-dimensional structure of the protein is altered. So, for example, let's say you have a protein, and this protein has, for example, an active site in this fissure. So what denaturation would do would kind of partially or completely unfold the protein so that there is no active site. So denat would produce no active active site, or as Deke says, the three-dimensional structure would be lost. 4. What usually distinguishes DNA from RNA? So A says strands are symmetrical. And, well, no, because symmetrical means that if you read the sequence, it would be the same if you, regardless of which position you started to read from. For example, the sequence would be the same if you read from the 3' and the 5', and that's not true. And here it says strands are anti-parallel, but there are RNAs single thread, so this is wrong. B. Contains adenine, contains cytosine. And yes, DNA does contain adenine, and RNA contains cytosine, but it's also the vice versa is also true. So DNA also contains cytosine, and RNA also contains adenine. So it's not a good way to distinguish them. So therefore, this is also wrong, because it's not something you could use to distinguish them. If you had said thymine uracil, on the other hand, you could distinguish them, because RNA has uracil and DNA has thymine only. C. Pentoses linked to phosphates, and like this is the basic structure of DNA and RNA, so it's not good to use to distinguish them. D. DNA double-stranded, RNA single-stranded. Yes, this is of course the correct alternative. In question 5, it says, research has shown that the genetic code is not entirely universal. Which research finding has shown this? And then first we have to analyze, what does it mean that a genetic code is universal, and what is the genetic code? Okay, so the bases in DNA are organized in codons. So for example, let me show you here. I will paste an image. So this image, Creative Commons, so I can use it in this video. It shows the genetic code. Okay, and you, if you start the code with guanine, G, because here it says 5', so, and that's the way you read DNA, it's always from 5 to 3', and then C, and then C or U or G or A, you get alanine, okay? So basically it means G, C is always alanine. And what is meant by universal is that this is the same for all species, and that means universal. Okay, so let's look at the alternatives and see if we can find out the right one. So some some amino acids are coded for by more than one codon, and this is this does not violate universality, because if you see here G, guanine, cytosine, and then cytosine or uracil or adenine, like the last base in this particular codon does not matter. So what it means is that you can have GCC or GCU. Both of them would give ALA, but as long as GCC and GCU means alanine, it's still universal. Okay, so this is wrong. So this is not evidence for breaking the universal code. B. There are differences between the base sequences of genes in different species. And this is also wrong. And the reason is that if two species are different, it's because of anatomical, physiological, and morphological differences. And those differences are due to different proteins. And different proteins means different amino acid sequences. And that again means different DNA. So to have a different species, you need a different genome. So that's something else entirely. C. In some organisms, the genetic code for mitochondria differs from the genetic code for the nucleus, and that is correct. So this is the right one. And so in eukaryotic cells, you have actually DNA in 2, or in plants 3 places. So, but in many animal cells, you have DNA 2 places. So you have some circular DNA inside the mitochondria, mitochondrial DNA. And then, of course, in the nucleus, you have the chromosomes. And in mitochondria, if you have adenine, uracil adenine, AUA, in mitochondria, so mito, it is coded for it is the code for methionine, but in the nucleus it is code for isoleucine. Another example is that in the mitochondria, UGA encodes tryptophan, but in all other many other species it is the stop codon. Okay. Number 6. This is a sequence of nucleotides from a section of messenger RNA. So we see there is no thymine here, and that is promising. That's good. But it's asked for which DNA sequence has it been transcribed, and then we can just pair these bases. So remember from which DNA sequence. So this adenine on mRNA must have been a thymine on DNA. It couldn't have been a uracil because this mRNA was made from DNA, it says here. And this uracil was made from the anti-coding strand, an A on the anti-coding strand. So the A coded for the U on the mRNA. And then this G must have been a cytosine, and this adenine must be again a thymine, and this must have been a thymine, and so on. And we see that this start only corresponds to TAC TT, this one, because we see that, for example, here C, it's like this mRNA was made from another mRNA, because instead of thymine you have uracil, but you don't have uracil on DNA, so it must be wrong, unless it was some kind of freaky virus that has some mRNA replication or something. So D was the correct one. Which gas produces most of the bubbles in bread dough? And here we have a rising dough. So the dough starts like this, and you have yeast in it, which is belong to the fungi kingdom, the fungi, and then it rises because these fungi, they metabolize the sugar in the dough or carbs and then make gases. And if it's aerob, you have oxygen plus sugar goes to carbon dioxide and water vapor, okay? And then if you have anaerob, you have sugar, and anaerob means no oxygen, so there isn't any oxygen here, goes to ethanol and carbon dioxide. And in practice, there are some oxygen that diffuses into the dough, and it will be used by the yeast. But most, I think most of the metabolism here is anaerobic. But anyways, we see that even if it's a 50-50 mix, we see that carbon dioxide is made from both processes. So regardless if it's aerob or anaerobic respiration, we get carbon dioxide. So here it asks which gas produces most of the bubbles. So then, of course, it's carbon dioxide. That meaning C is the correct alternative. What is the process shown in this image? And of course, we can see that it's dividing. Okay. But what cell is dividing? Well, it has no organelles, that we can see at least. And then, if we look here, especially here, it looks like the cell wall is, that there is a cell wall. Like it's not a thin membrane, but maybe even a cell wall and a capsule. You can see it's kind of like bilayered. This is not, at this resolution, you cannot see the phospholipid membrane at this. The phospholipid membrane would just be a thin line. So probably cell wall plus maybe a capsule. Or maybe it's a gram negative, which has a kind of a double wall. So no organized. So this means it's a prokaryote. And the question is, what process is shown? Well, a binary fission of a prokaryotic cell is then indeed correct, because cell division in prokaryotes are called binary fission. So this is correct and this is correct. B is wrong because it refers to an eukaryotic cell and meiosis, which only takes place in eukaryotes. C is wrong because it refers to mitosis, which is a eukaryotic phenomenon. For example, you need a nucleus to have mitosis, and chromosomes, of which the prokaryotes have none. And here again, it refers to eukaryotic cells. So all of these are wrong. 9. What distinguishes prokaryotic cells and eukaryotic cells? So prokaryotic cells have a cell wall, yes, and eukaryotes does not, unless you are a plant. So yeah, maybe we can put the red under it. Yeah, because plants are eukaryotes and they do have a cell wall. Eukaryotic cell has a plasma membrane, but also prokaryotes have a plasma membrane. So I think we need to just take away option A. I think it's wrong. B. Prokaryotic cells. Cell structure not compartmentalized, and that is true. And all eukaryotes have compartmentalization. For example, in the nucleus, which stores all the chromosomes. So this one is probably the correct one. But let's just check the two others. C. Smooth endoplasmatic reticulum. No, prokaryotes don't have this organelle. So this is wrong. And D. No ribosomes. Of course, prokaryotic cells do have ribosomes, and some define ribosomes as an organelle. And if so, then the ribosome is the only organelle prokaryotes have. So this is also wrong. 10. What can be deduced about a striated muscle fiber from both of these statements? A eukaryotic cell has one nucleus, and we can see this is typical cell theory. Like cells have certain common features, and one nucleus per cell is one of those features. A striated muscle fiber has many nuclei, and this is also correct, but it's an exception to cell theory. So what can be deduced about the striated muscle fiber? Let's say it is prokaryotic. No, because prokaryotes don't have nuclei. B. It is an exception to cell theory. Yes, this is the correct one. It consists of aseptite hyphae. Well, yes, some fungi have multiple nuclei because they don't have sectioned off their hypha segments, but it doesn't have anything to do with striated muscle fibers. D. It's preparing to divide. And not necessarily. There is nothing here which, like, it's not like muscle cells divides to begin with, or that they, at least I don't think they divide after they are fused together, but I'm not sure about that. But anyways, it has nothing to do with cell theory, so these two are wrong. 11. The diagram is a model of one type of movement across a membrane. And we can very easily see that here on this side, let's say it's the inside, you have high concentration, and here on the outside you have low concentration. And we see also that these let's think of them as ions. We don't know if they are, but let's just think that they can't go through here, because if they are polar, they won't go through the hydrophobic core of the phospholipid bilayer membrane. Like these fatty acid tails are really hydrophobic and hates everything polar. So it goes through here, and this is of course diffusion, first of all, since it goes along the concentration gradient. Along concentration gradient. And why is it along the concentration gradient? Because it's lots of molecules here, very few here. And that permits passive transport, like transport of molecules without using energy. That is only possible along the concentration gradient. So it is, of course, facilitated diffusion. Why isn't it simple? Because it doesn't go through the membrane. Like some polar molecules could maybe go through membranes at a very slow rate, so that would be simple diffusion. Like water sometimes go through the membrane with simple diffusion, even though it is a polar molecule. Osmosis could maybe have been correct, but it's like yeah, it could have been correct, but then I don't think they would have shown it the molecular circles. Like this looks more like an ion. And also only aquaporins permit water to go through, but water can also pass through the membrane. So osmosis is not strictly wrong, I think, but it just doesn't fit very well, so we cross it out as well. And then the active transport, that's wrong because no ATP is used here, and anyways you don't need active transport to transport stuff along its concentration gradient. 12. The statement relates to Pasteur's experiments. So he demonstrated that if broth, so broth is kind of like a nutrient-rich fluid. So if broth was boiled to kill all organisms and was kept in swan-necked flasks, so they were, these swan-necked flasks were like this, and then they had like this swan neck, like went like this, and then there was water in this neck. So this water was like a seal, because if like there was no passage of air through this water lock, so to speak. So if it was kept in swan-necked flasks, preventing the entry of organisms, no organisms grew in the broth, and the broth was here. Maybe it should be green. I don't know. We need to make it brown. Broth is brown. So there was no organisms growing in the broth in the flask. But if the swan-necked flask was broken, mold soon started to grow in the broth. So if we broke the flask, then air could go directly in, and air carried spores, and then the spores started to grow in the broth. Okay, so what did this statement suggest? One, mold evolved by endosymbiosis. No, we We have no evidence to suggest that. Oxygen is required for anaerobic respiration. No. Like, even if Pasteur removed the oxygen, mold could grow in his broth. And if it was oxygen, then the mold could still grow. Molds can often grow both anaerobic and aerobically. C. Cells can only be formed from division of pre-existing cell. And this is indeed the correct thing. And this might seem a little bit strange if you didn't read topic 1 very well, but in the old days, people believed in something called spontaneous generation, meaning they thought bacterial growth just arose spontaneously. And Pasteur showed no, that is not the case. If things are sterile, nothing will grow, not even bacteria. 13. What event occurs only in meiosis? Fusion of gametes to promote genetic variation. So fusion of gametes happens after meiosis. First you make the sex cells, then you see if some of them find their counterpart. So that's one. B. Random separation of chromatids. And, well, this is a hard one to rule out, but it is wrong. I can cross it out. Because chromatids are technically randomly separated in mitosis also. You know, mitosis is in normal body cells. Because, let's say in mitosis, you have chromosome, let's say chromosome 1. And here you have the father version, the one you got from your father. And here you have the mother version. And during mitosis, these are situated along the equatorial plane of the cell. And then they are being dragged by spindles to each phase. So in any phase, they will be dragged like this. And so kind of both the father chromosome and the mother chromosomes are drawn to each cell so that you make 2 identical copies. But if you are somehow able to mark this chromatid, then you would see that it would be random if it went to this cell, but it doesn't matter anyways. So that's why this is wrong. Because, well, technically, chromatids are separated both in mitosis and meiosis, even though it doesn't matter. C. Random separation of homologous chromosomes, and that is, of course, the correct alternative. And it's because here in mitosis, let's say this is mitosis, and this would be random. It is 2n. Because you only have the separation of the sister chromatids, but in meiosis, then you separate the 2 chromosomes. So one chromosome is dragged along to one cell and the other to another, so that now each cell becomes n, and each cell has only one version of the chromosome. It's now haploid, and one specific cell for a specific chromosome only have the mother chromosome, the one you got from your mother or the one you got from your father. 14. Is it possible for 2 parents to have children with each of the 4 ABO blood groups? What blood groups would the parents have? And the answer is yes, it is possible for 2 parents to have children with all blood groups. So when we write this, you can either write it like this. So you do I and then A. This is kind of like the gene for blood type I, and so on. But let's just use A and B's, and O would be kind of the I without anything, and this would be capital B. Okay. So what blood types in the parents would be necessary to produce all blood types in the offspring? And let's just draw a Punnett grid, an experiment. So let's say the mother is heterozygous, so she has A. Now I'm using this notation, okay? Let's say the mother has I and O, okay? And let's say the father has B and O. And then we see that both blood types are possible, because then the children would have AB, BO, and then AO and OO. And we see that this is blood type, this one is blood type AB, this is blood type B, blood type A, and blood type O. Okay, so this is more like phenotypes. Let me just write it, the genotypes properly. So I, A, I, B, I. So if you are like a stickler for genotypes, it would be, this would be I, A, I, B, I, B, I, and then I, A, I, and I. So this would be actually blood type O. So we have created the 4 genotypes. So so is it possible for 2 parents to have children with each of the 4 blood types? So we see that we have blood type AB here, AB. We have blood type B here. We have blood type O here. We have blood type A here. And what did the parents have to be able to make this combination? 25% of each. It's easy. Blood type B. So that's the phenotype. The genotype is actually BO. Or using the other notation, IB, I. And blood type A. And we see that these 2 alternatives correspond to alternative D, which is the correct one. 15. What is the effect of dominant alleles? Well, they mask the effect of recessive alleles, and that is correct. For example, if you have the gene for brown eyes and blue eyes, then you will be brown-eyed because the brown gene completely masks the expression or the phenotype of the blue one. So this is correct. 2. They become more frequent than recessive alleles in a population. No, this would have to do, the evolution would determine this, not dominance. So of course, because a dominant gene more often produces its phenotype, you would see it more, but it wouldn't become more frequent as a gene or allele. 3. They have a joint effect with recessive alleles when characteristics are codominant. And this is also wrong, because they only have a joint effect with other codominant genes. So here only A is correct. 16. Which techniques separates proteins according to size? And A is treatment with restriction endonucleases, and this is wrong because this doesn't have anything to do with proteins. This is a way to cut DNA. So restriction enzymes, those are DNA, and nucleases, you hear that it do something with nucleotides, which are DNA or RNA. So this is wrong. B, PCR, that's polymerase chain reaction, and it's used to not to separate something but to copy something, and that something is DNA. So it's used to make lots of copies of DNA, so it doesn't have anything to do with proteins. C, gel electrophoresis, and that is of course the correct one. And gel electrophoresis, then you have a gel like this, and then you have some holes in the gel, and you put your proteins in here in these openings, and then you have I don't remember which had a positive charge and which the negative, but let's say it's positive here and then negative here, and then I think actually it's opposite, because I think the proteins are treated to become negative, so it's negative here and positive here, and then the proteins kind of travels like this from against the negative pole. But the thing is, if you have a long protein, it will move slowly in gel, so a long protein might just go to here and stop, but a short protein will maybe go all the way to here, because a short protein doesn't have that much friction in the gel. 17. Which category of organisms is correctly described by its method of nutrition and site of digestion? Okay, so consumer is a heterotroph, and that is correct. So so a consumer is indeed a heterotroph, because heterotroph just means you get your energy from living carbon or newly dead carbon. So for example, if you eat a sheep, you are heterotroph. You get your energy from that meat, and also the building materials. Site of digestion, internal, yes. For example, you have an alimentary canal to digest the meat. But a consumer could just as well eat grass or fruit or something. Then you would be a primary consumer. If you ate sheep, then the sheep would be the primary consumer, so you would be maybe secondary consumer. B. A saprotroph is also a heterotroph. So a saprotroph is not heterotrophic. For example, a saprotroph can be fungi, which have hyphae that grow through the food. For example, if you left some bread to mold, and the fungi absorbs the nutrients across its whole body. So site of digestion is indeed external, but this alternative is wrong because it claims saprotrophs are autotrophic. That is only the case for photosynthesis. C. Producer is autotrophic, yes, but then it says site of digestion, internal. And well, producers such as grass or algae, they don't have much of digestion at all. So this whole column is not very well placed, because you can say that chlorophyll digests sunlight. No, it doesn't work. You need bosons to digest something, and sunlight is muons. Okay, so this doesn't work. Maybe we have to do like this, since the whole category is wrong. D. Detritivore is heterotrophic, yes, but the defining feature of detritivores like woodlice, have you seen woodlice? They look about like this. Antennas, they crawl. If you kind of take up a log, you often see them. And they crawl around, and they have internal digestion, not external. So detritivores are very similar to saprotrophs, but saprotrophs absorb the nutrients across their body, and detritivores digest them in an alimentary canal. So the correct one here, the only correct one was A. 18. What limits the length of food chains in an ecosystem? And like the size of individual organisms does not really limit it. The competition between organisms does not limit it. Like, for example, what actually the best alternative here is the loss of energy between trophic levels, because you could argue like this with the size here, that the blue whale is really big, and that's why that food chain is very short compared to other marine food chains, but C is better. And the reason is that the loss of energy is so efficient in reducing the length of food chains, because the loss of energy is 90%. So if this one is 100 gigajoule or something, then only 10 gigajoules are available for the next, and then only 1 gigajoule, and so on. So I would say C is the correct one. 19. How can mollusks and Plathiel Mintus be distinguished? So a mollusk is like, for example, a snail, and it does have a mouth here, and I think it has anus somewhere here and in the back. But Plathiel Mintus, they have a feeding tube, and then they have this strange internal kind of digestion sac with no anus. So all wastes, they go kind of through the body, so they have no anus. So the correct alternative here is B. Mollusks have a mouth and an anus, but Plathiel Mintus do not. They only have a mouth. What is a feature of shorter wavelength visible radiation? And A is indeed correct. It says it includes violet light. And violet is actually the shortest wavelength we can see. And B is wrong. It says it has less energy per photon than longer wavelength. Actually, energy is inversely proportional with wavelength. This is because it's actually frequency that is proportional to energy. And frequency and wavelength is inversely proportional because their product is always the speed of light. And speed of light is always the same. C. It is absorbed by greenhouse gases. And that is also wrong, because it is actually longer wavelength that is absorbed by greenhouse gases. So if we have the Earth like this, then you might have short wavelengths like blue light, which heats up a portion of the Earth, and then long wavelengths are returned, but those are kind of reflected back by greenhouse gases such as ozone, and it goes back to the Earth. So this is also wrong. D, it's reflected by chlorophyll, and that is also wrong, because if you have a leaf, then we see it as green because the green light is actually reflected and seen by the eye, but red light, but interestingly for this, in this situation, blue light is both absorbed and used in photosystem 2 and 1. 21. Which is a coniferophyte? And here they say Addissonia digitata, an African baobab tree with white flowers. And flowering trees, those are often leafy, and so I don't think it's a pine or something, which conifers are. B, Cythia australis, an Australian tree fern producing spores. So that does not sound like a pine. And then Hypnum plumiforme, a green plant with no vascular tissue grown in Japanese gardens. So no vascular tissue means basically a moss, a bryophyte, and that's also not a coniferophyte. And then D. Pinus strubus, a North American tree with ovules on scales that are not enclosed in the ovary. And that is, of course, a coniferophyte, so this is the correct one. And what is this ovules, ovule tuck, and so on? And that's just that, for example, a flowering plant, they have ovule here. That, like this part here is the ovule. But that also is the case for the cone in coniferophytes. So it has kind of the ovule here. Let me see if I can ring it for you. It's very small. But what the cone doesn't have is the ovary, this whole protective. So it doesn't have a thing going around here. Like it's no space for it. Yeah. So what it basically means is that the coniferophyte seeds often have less kind of germination energy than what flowering plants can offer. 22. Which process promotes variation in a population? So A, mutation, that is correct. So mutations are random changes in the genetic code. And those can, for example, change the codon, meaning another amino acid is inserted into a protein, and that can give that protein different features or functions, and that again can give an evolutionary advantage. It can. It doesn't have to, but it can. Question 23. So here we see the skeletons of a frog and a rabbit, and the femurs are indicated with X and Y. And the question reads, what is the evolutionary relationship between X and Y? So alternative A says they are analogous. And what that means is that you had different structures that had a different origin, but because of convergent evolution, they evolved to become the same structure. They were shaped by the environment during the process of evolution, so to speak. But that is not the case here. So, because we know that the tetrapod, or a creature going on four legs, descendant from the bony fishes, is actually the common ancestor of all vertebrates, and it already had the hind leg. It had one pair of forepaws, one pair of hind legs, and so this is the same structure, and it has always been used for locomotion or legs. So these structures are homologous, meaning they were used as legs in the common ancestor, and are still used as legs. 24. What happens to starch in the small intestine? And starch is a polysaccharide. So alternative A is wrong because it says endopeptidase. Ase means enzyme, and peptid means polypeptide. That it breaks chains of polypeptides, meaning proteins. That's not carb. B, contraction of intestinal muscle mixes starch with enzyme. Okay, sounds like a good start. To accelerate its conversion, yes. Into amylose, no. Amylose is one of the components of starch. And so, yeah, you cannot make it into something it already is, for the first. For the second, the body wouldn't want to convert one polymer, amylopectin, for example, into amylose. it's still starch, it still can't be taken up by the epithelium. And thirdly, digestion is about making monomers, not converting one polymer to another. C. Glycogen secreted by the pancreas hydrolyzes starch into glucose, which is eventually transported to the liver. And well, the pancreas do secrete digestive enzymes, but not glycogen, because that is the storage polymer in the body. So yeah, but this part is correct. Hydrolyzing starch into glucose, that works. And glucose is indeed transported to the liver, but not eventually. Like the portal vein goes directly from the small intestine to the liver to kind of protect the body from the fluctuating levels of glucose after digestion. So then it must be D. But why is it correct? Amylase secreted by the pancreas digests starch to enable absorption by the villi. Yes. So amylase, well, you can hear that it's an enzyme because it ends with ase, and it then break down amylose. Yes. Amylose is a part of starch. Yes, so everything checks out. And yes, it is indeed secreted by the pancreas, the major digestive juice gland in the body. And it is indeed, the monomers produced are indeed absorbed by the villi, this protrusion from the small intestine that increase the surface of the intestine manifold. 25. The diagram shows the layers of the tissues in a transverse section of the human small intestine, which is the layer labeled with the letter X. And that is the circular muscle, because out here you have the longitudinal muscles. Enal, is it? And X is the circular muscles, and then A is the mucosa, and then you have the villi here. 26. Which discovery was an indication that the heart pumps blood to the body through arteries? And A says the amount of blood pumped exceeds that of blood produced. Well, that the blood is reused and it's produced very slowly in the bone marrow, so it may be true, but it doesn't indicate the direction. B. Blood could easily be pushed up a limb vein, but not down. And here is like, what does up mean? It's quite arbitrary, so. C. The observation that there were pores between the right and left atria. And, well, in humans, we have a closed heart. I think in some animals, the heart is open. But anyways, it doesn't show where the blood is pumped. So the correct alternative here is the heart swelled up when the arteries were tied in an animal experiment. And the reason that the heart swelled is that all the internal valves in the heart is one way, pointing towards the arteries. So if you clamped the arteries, then the blood would have nowhere to go because it wouldn't go back because of the one-way valve. So this seems very true. So 27. Which is a valid comparison between arteries and veins? And as you know, arteries, they transport blood from the heart. So that means thick walls because of the high pressure. But veins, they transport blood back from tissues to the heart. And so they don't need to endure that high pressure. And also they have kind of these one-way valves built in that helps blood go in the right direction. So alternative A is arteries carry oxygenated blood. And that might seem correct. They carry blood from the heart, but you have to remember there are two circulations. So if we make a heart here, then you have the large circulation to the body tissues, and those are indeed oxygenated blood. So we can maybe make a red here to indicate it's oxygenated, since the heme group is a little more red when it has oxygen. And then deoxygenated blood is pumped back through the veins. So that's okay, but in the small circulation you have also you send deoxygenated blood to the lungs through arteries. So here the arteries transport deoxygenated blood, and then the veins transport back the oxygenated blood. So these are indeed, this is wrong, and also we see that veins indeed carry oxygenated blood from the lungs, so this is wrong, so A can be ruled out. B, return blood to the atria of the heart. That's the job of arteries. And well, arteries never return blood to the heart. So arteries, they go from, let me show you. So let's say these are the atria, and these are the ventricles. So the blood, so the arteries, they only transport from the ventricles. Let me show it here. So here you have the left ventricle, it transports blood through the aorta, really thick vein that comes like this, and goes around like this, and have a lot of branches to the heart itself, to the upper body and brain and so on, and then it goes down here, and then it supplies the underbody here. Okay, so the left ventricle, for example, transport blood from the heart to the tissues, so never back. So this is wrong. It doesn't return blood to the atria, it transports blood to the tissues. And then veins, blood flows from the ventricles to body tissues. No, that's arteries, so B is also wrong. C, blood flows at high pressure. Yes, this is indeed true, because since the arteries always take the transport from the heart, they endure the highest pressure. So that is correct. And veins, blood flow at low pressure. Yes, and that's why they need these built-in one-way valves, because veins have to do this job at lower pressure. Let's look at D just to be sure. So D says arteries have thick walls. Well, yes, they do. Veins have permeable walls. No. So neither arteries or veins are very permeable. The reason for this is that the transfer of gases and waste molecules and nutrients are performed over the capillaries, and the capillaries are in the body tissue. So in the body tissues you have this network of capillaries, which are fueled by the arteries and drained by the veins. 28. The diagram shows changes of pressure during the cardiac cycle. And we have three phases, 1, 2, and 3. And then you have pressure here on the y-axis, and then time along the you have pressure along the y-axis and time along the x-axis. And it says, which is an explanation of the processes happening. And then, of course, they are talking about this cardiac diagram. A. During phase 1, pressure in the left ventricle decreases while it increases in the aorta during contraction of the left atrium. And this seems wrong because, again, here is a very simplified version of the heart. And then we have to divide into the atria and left and right ventricle. And this left ventricle, which of course is on the right side because we watch it from the outside, but in the body this would be on the left side since we watch at it from the right. When this left ventricle contracts, it flushes blood out into the aorta, which goes like this. And when this happened, the pressure is high both in the ventricle and in aorta at the same time. Like, it wouldn't make sense. So during phase you can't have decreasing pressure in the ventricle and increasing aorta. It's impossible because they are connected during the stroke. And anyways, it's the wrong phase. So during the phase 1, we see that the ventricle is filled by the atrium, because you see the atrium starts squeezing blood. So this is the left atrium, and it has this channel, one-way channel with a valve that only permits the ventricle to be filled. And so you see that the left atrium first started to squeeze because the pressure increases, and then the pressure inside the ventricle increases a little bit, but it's just the filling of the ventricle, this part here. Ventricle. Okay, so this is so A is all sorts of wrong. B, in phase 2, pressure rises in the aorta because the left ventricle is contracting. And here I think everything is correct, because if we look at the left ventricle, maybe we should draw it with yellow. So here is the left ventricle. So here it starts to get filled by the atrium, yes. And now here the heart fires, the atrioventricular node fires, and the whole ventricle contracts explosively. And we see that the pressure in the ventricle, of course, increases when it contracts, and so does the pressure in the aorta. Here, I show that with red. So this is the aortic pressure. And why does it suddenly increase here? It's because at this point, the valve to aorta kind of opens, because, or it's not actually, yeah, it's a one-way valve. So aorta has kind of this one-way valve here, that this valve only opens when the pressure in the ventricle, left ventricle, is greater or equal to pressure in aorta. Okay, so then when blood finally manages to squeeze through this valve, then the pressure is similar in the ventricle and aorta, and it flows into the body. So I think this is correct, B. Everything here looks correct. Let's just check C in case. So in phase 3, pressure decreases in the aorta because the left atrium is relaxing. Okay. In phase 3, so this is the gray one. Pressure decreases. Okay. Well, it does decrease in the aorta. Yeah, it decreases a little bit here. Okay. And why do they say this happens? Because the left atrium is decreasing. Well, this is wrong because the left atrium are not directly connected with the aorta. Look, the left atrium gets blood from the vena cava, so it has nothing to do vena cava, like the main vein. It has nothing to do with the ventricle, so C is really wrong. D. In phase 3, there is a constant increase of pressure in the aorta, as both the left atrium and ventricle are at rest. And this is also wrong because we can very easily see that the pressure in the aorta in phase 3 gradually declines. And it will until the ventricle, left ventricle, again fires. 29. Which statement is true for the antibiotic penicillin? Watson and Crick developed its usage, and that is wrong because I happen to know that Watson and Crick, they discerned the structure of DNA. B. Penicillin blocks processes unique to eukaryotic cells. No, it's the other way around. Penicillin, they actually block processes in bacterial cells. That's why we like to use it to treat bacterial infection. C. Viruses lack metabolism. Well, yeah, that's true. Viruses, they take over the machinery of a cell and use the cell to make copies of itself. So that's correct. And penicillin has no effect on them. Yes, on the virus itself, penicillin has no effect. So this is true. D. Florey and Chain sequenced the genome of penicillin notatum, and these people did not live in a time, or they weren't active at the time when genome sequencing was available. 30. How can knowledge about the pineal gland function be applied? Yeah, so the pineal gland is the gland that controls circadian rhythm in many animals, or most animals, and it is controlling the release of the sleep hormone melatonin. So if you have the brain here, then the pineal gland is kind of back here. So very small but very important. To restore sleep time by the use of melatonin. Yeah, this sounds very correct because melatonin, the pineal gland releases melatonin. Melatonin affects sleep time. Yes. B. To trigger ovulation during in vitro fertilization. No, that doesn't sound correct. C. To reduce sperm production in male contraception. No. To regulate blood sugar in type 1 diabetes. No. That sounds more like insulin. And that was the last question. So in general, if I'm not entirely wrong, is it the 19th you have the exams in 2021? Okay, so my advice to you is when you do the exam, take time to kind of organize your knowledge by drawing. Okay, and what should you draw? Like, organize your knowledge if you have a difficult question, or maybe make Venn diagrams and stuff like that. Also, look over the command words. Okay, that's not that relevant for paper 2, but paper 1, but especially for paper 2. And read questions carefully. Okay, so good luck on the exams.